Hello everyone, welcome to my channel Anupma Biology Classes. In this video, our topic is important terms of the photosynthesis. This video is taken from Plant Physiology series, CBSE class 11th, part 2nd of chapter 14. Now, let's see here the lecture overview. So in this video, our topics are nature of light, absorption spectrum and action spectrum, two steps of the photosynthesis, that means light reaction and dark reaction, two pigment systems in which photosystem first and photosystem second, and difference between photosystem first and photosystem second. Now, let's start the topic in which our first topic is nature of light. Solar radiation, according to electromagnetic wave theory, which is proposed by James Clerk Maxwell in the 1860, can be described as photons that travel in waves. The unit quantity of the light energy in the quantum theory is called quantum, whereas the same of the electromagnetic field is called photon. Solar radiation can be divided on the basis of wavelengths. Radiations of the shortest wavelength belong to cosmic rays, whereas the of the longest wavelength belongs to radio waves. So, the different radiations on the basis of increasing wavelength and the decreasing energy per quantum are listed as cosmic rays, gamma rays, X-rays, ultraviolet radiations, visible light, infrared light, microwaves and radio waves. So here you can see that the electromagnetic spectrum, the visible spectrum of the radiant energy lies between 380 to 780 micrometer. Solar radiation that hits the Earth atmosphere, about 42%, undergoes major modifications. A major part of it is filtered through the various constituents of the atmosphere, that is, high energy radiations are screened out by the ozone layer in the atmosphere, and lower energy radiations are screened out by the water vapor and carbon dioxide. Our atmosphere thus provides a window through which a visible wavelength pass on to the various life forms of the biosphere. Now here you can see that visible light. It is the portion of the solar radiation which occurs our eyes can see. It consists of radiations having a wavelength between 380 to 760 nm. The biological systems make most effective use of the visible radiations. So here you can see the use of the visible indigo. First one, that is 430 to 470 nm, then blue 470 to 500 nm, then green 500 to 580 nm, yellow 580 to 600 nm, and the orange 600 to 650 nm, and red 650 to 760 nm. So these are the visible light which can further resolve into the light of different colors, and these colors are violet, indigo, blue, green, yellow, orange and red. Now, after this, absorption spectrum. What is absorption spectrum? Absorption spectrum of a particular pigment is the curve plotted on a graph paper representing the amount of light absorbed at each wavelength by that pigment. To determine the absorption spectrum of a particular pigment, a purified sample of that pigment is placed in the path of a visible light inside an instrument called a spectrophotometer. This instrument measures the amount of light that passes. Here in this picture you can see. So the amount of light passes through the pigment solution, and from this it can be calculated how much light of a particular wavelength was absorbed. Then the result is plotted on a graph which represents the absorption spectrum of that pigment. The absorption spectrum of chlorophyll A and chlorophyll B, here you can see this picture. The figure shows that these pigments absorb maximum in the blue and red region of the visible light. The chlorophyll A molecules absorbing maximum at different wavelengths are often known after the wavelengths they absorb a maximum. For example, chlorophyll A 670, chlorophyll A 680, chlorophyll A 700, etc. So this is the absorption spectrum of the chlorophyll A and the chlorophyll B. Now, after this, action spectrum. So what is action spectrum? Action spectrum is a curve plotted on a graph paper representing the amount of oxygen evolved or the amount of carbon dioxide fixed or any other action of the photosynthesis at different wavelength of light. It has been observed that the photosynthesis occurs maximum in the blue and red regions of the visible light. It is possible to determine the relative contributions of a particular pigment in the process of photosynthesis by comparing the absorption spectra with the action spectra. In case of higher plants, the absorption spectra of the chlorophyll A and chlorophyll B run almost parallel to the action spectra of the photosynthesis, depicting their roles in the process. So here you can see that the absorption spectra of the chlorophyll A and the action spectra of the photosynthesis. In action spectrum, absorption of the absorption spectrum of chlorophyll A and B, and at the upside you can see that the rate of the photosynthesis, that is the action spectrum. So in this picture, both are present. That means absorption spectrum and action spectrum. Now, after this, two steps of the photosynthesis. So what are the two steps of the photosynthesis? The present state of our knowledge regarding photosynthesis proves that it is not a one-step process, but it is a two-step process. The first step is dependent on light and hence called the light reaction or the photochemical reaction. In this step, the assimilatory power is generated in the thylakoids. This step is followed by the second step, in which the light is not directly required, but the assimilatory power generated in the light reaction is used up for the fixation of carbon dioxide into the carbohydrate. This second step is called dark reaction or the thermochemical reaction, which takes place in the stroma of the chloroplast. So these are the two steps. First one is the light reaction and the second is dark reaction. Light reaction, where the light is absorbed by the chlorophyll, energy from the light splits water, which we will discuss in the next video. Now, after this, two pigment system. So what are the two pigment systems? Light reaction of the photosynthesis involves the participation of the two separate pigment systems of the photosystem, that is photosystem first, pigment system first and the pigment system second. Each photosystem has a pigment protein complex composed of a chlorophyll A, chlorophyll B, carotenoids and other components required in electron transport. Each pigment protein complex consists of a core complex and an antenna complex. Various pigment molecules in the antenna complex absorb different wavelengths of light and transfer their absorbed energy to the core complex, which is finally concentrated in the reaction center chlorophyll. The latter takes part in the photochemical reaction. The role of antenna complex and the core complex is significant in harvesting the maximum solar radiation over a large area and passing their absorbed energy to the reaction centers. Now, in photosystem first, the photosystem first complex contains a single molecule of chlorophyll A700 or P700. This single molecule is the reaction center for the photosystem first, and to which all the necessary pigments molecule in this photosystem transfer their energy. Besides P700, the photosystem first consists of 200 to 400 chlorophylls, 50 carotenoids, and 2 iron-containing proteins similar to ferredoxin called FES protein. The FES protein are the primary electron acceptor of the photosystem first. So, the pigment system first has more chlorophyll A molecules as compared to chlorophyll B molecules and carotenoids. It is light green in color and located both on the non-appressed part of the grana thylakoids as well as on the fluid channel, mostly toward the outer surface of the membrane. Photosystem first is associated both with the cyclic and the non-cyclic electron transport and drives electron from photosystem second to NADP+. However, it can carry on cyclic electron transport independently. Now, photosystem second complex contains single molecule of chlorophyll A680 or P680. This single molecule is the reaction center for photosystem second and to which all the accessory pigment molecules in this photosystem transfer their energy. Photosystem second also contains about 200 chlorophylls and 50 carotenoids except the reaction center. All the other pigment molecules are called accessory pigment molecules. Photosystem second also contains its primary electron acceptor called pheophyte. So, besides pigment molecules, the photosystem second also contains quinone, which quenches the electron from P680, manganese-bound proteins, and an unknown water oxidizing enzyme. So, the photosystem second has almost equal number of chlorophyll A and chlorophyll B molecules. It is dark green in color and located mostly in the appraised part of the grana thylakoids towards the inner surface of the membrane. Photosystem second is associated with the non-cyclic electron transport, splitting of water, evolution of molecular oxygen and reduction of NADP+ to NADPH. So this is all about the pigment system. After this, now what are the main differences between the photosystem first and photosystem second? In photosystem first, photolysis of water is not occurred, while in the photosystem second related to with the photolysis of water. In photosystem first, reaction center is P700, while in case of photosystem second, reaction center is P680. Photosystem first is rich in chlorophyll A then chlorophyll B, while the photosystem second is rich in chlorophyll B then chlorophyll A. Photosystem first, molecular oxygen is not evolved, while in case of photosystem second, as a result of photolysis of water, molecular oxygen is evolved. Photosystem first receive electron from the photosystem second, while the photosystem second receive electrons from the photolytic dissociation of water. In photosystem first, pigments absorb longer wavelengths of light, while in photosystem second, pigments absorb shorter wavelength of light. In case of photosystem first, in this reaction NADPH is formed, while in case of photosystem second, NADPH is not formed. Photosystem first can participate in both cyclic and non-cyclic photophosphorylation, while the photosystem second just participates in non-cyclic photophosphorylation. In photosystem first, the core complex is composed by a smaller number of protein, while the photosystem second, the core complex is composed multi-subunit of about 25 to 30 subunits. Photosystem first lies on the outer surface of the thylakoid membrane, while the photosystem second lies on the inner surface of the thylakoid membrane. Photosystem first has iron-sulfur type reaction center, while the photosystem second is a quinone type reaction center. The major function of the photosystem first is NADPH synthesis, while the major function of the photosystem second is hydrolysis of water and ATP synthesis. So these are the main differences between the photosystem first and photosystem second, and this is the end of video in which we knew about the photosynthetic synthesis, action, nature of light, absorption and action spectrum, steps of the photosynthesis, pigment systems and their differences. So the next upcoming video will be on the mechanism of the photosynthesis, in which the first one, that is the light reaction. So this is the end of video. If you understand this video, please like and share it and subscribe to my channel Anupma Biology Classes. If you have any questions, any queries or any suggestions, you can ask in the comment section below. Thank you for the watching.